So, should I wait or get started?
It's time.
Okay. Alrighty. So, my name is Negacy. I'm from University of Colorado. Today, I'm going to talk about one of my projects. Its title is Biomedical Concept Normalization using sequence LSTM model. LSTM stands for Long Short-Term Memory. First, I would like to mention people involved in the project. My mentor, Dr. Larry Hunter, sitting right there, and our ontologist, Mike Beda, and my collaborator, Asman Lash. So I'm going to start by defining what the task is. So basically, in this task, what we wanted is we want to find biomedical terms and assign them unique IDs. So here I'm showing you a sentence, and the right there, those are the annotations. So, for example, here we have sequence ontology. SO stands for sequence ontology. I'm going to talk more about the ontologies. And we have protein, again, sequence ontology. So, but most importantly, those terms are identified and assigned IDs. Those are the term IDs. And these are found from ontology. So, previous approaches have been mostly dependent on dictionary lookups, which means it's string matching, which has its own limitation. It doesn't capture ambiguity, but our approach is completely using machine learning. Here I have two steps. So given a document, what we want to do is find the textual mentions using CRF, which is conditional random fields. That is our first step. And once we found the terms, we can assign them unique IDs using the second step, which is sequence-to-sequence mapping. So our contribution is on the second step. The first step is kind of normal thing, but our novelty is right there on the second step. So here I'm showing you a sentence again. So the first step basically is going to find those mentions involved. So once we found them, again, on the second step, so for each of those, we wanted to assign those unique IDs. So we are doing this on our corpus. It's called CRAFT. It's developed in the Hunter Lab, University of Colorado. It has 3 versions. So on the first version, what they annotated is syntactic and semantics. So the semantics are basically the ontologies. So here they have annotated 8 of them. And on the version 2, they did coreference annotation. And on the third one, they have annotated 2 more ontologies, the molecular process ontology and the Oberon. In addition to those, our ontologies defined extension classes. So these are basically a composition of existing classes. So for this work, we will focus on the first version and the last one. So I will report my results in 3 different data sets, the version 1 and the version 3, which is the the proper object classes and the proper object classes plus the extension classes. So the task is not new. So concept normalization has been done like for years. Part of the reason why it is difficult is I would say there are 2 main reasons. The first one is from language perspective. Lexical variation and lexical ambiguity are the main issues. So when we say lexical variation, there are different ways to mean one thing. So those are really difficult to identify and map them. And again, a given term could have multiple meanings, or even English words could be confused with biomedical terms. So we have lots of ambiguity. And from methodology perspective, we have enormous classes, and it's really hard to get data annotated with all of them. So we are always prone to data sparsity. So these are the main reasons why the task is still difficult and unsolved. So our approach for the second step, again, most of my talk is for the second step because that's really our novelty. So our approach is like given the textual mentions or the biomedical terms, so we want to apply like sequence model for mapping them, and we want to produce the individual IDs. So here's an example of a biomedical term, and what we want is to predict its ID. So the way we model this is using character-based sequence model. So when you say character-based, our input is the characters, the individual characters in the biomedical text. the biomedical term. So for example, for the input developmental mechanism, we have an encoder and a decoder. So basically, the encoder is going to process each of the characters in the input, and it will pass it to the decoder. So in the decoder, we will predict each of the characters one at a time. So part of the reason we are predicting the individual digits is that we are approaching the task as a sequential task instead of just classification. If it is classification, we're not leveraging the information around those digits, but if it is sequential, for example, to predict the letter O, we will leverage the information before it. So in this way, we're approaching the task like a sequential mapping task. So similar approaches have been applied for different tasks. For example, for machine translation, they have been effective. They have been also applied for data normalization, and we're applying this for biomedical term normalization. So this is our architecture. It has 4 main components. Right there we have the encoder. It is a bi-LSTM. So we have a bidirectional LSTM. Basically, we wanted to process the information in both flows. And we have an attention mechanism. The decoder is right there, and we have the softmax. So here, for example, for the term developmental mechanism, so the first input is one hard code representation of the characters. So we have D right there. So we'll pass it to the encoder, and similarly, we will do the same for all the characters right there. The dimension of the bidirectional LSTM is the right there, that's TX. That's basically the maximum length of the input in our training set. The importance of the attention is, the idea behind this is that when you are predicting one of the characters, let's say when you are predicting the first character right there, all the characters are not equally important. So the attention mechanism will give different weights for each of them. It is basically a weighted sum of the weights, the activation weights you get from the bi-LSTM. And once we did the attention, so we have the decoder. Basically, the decoder will produce one character at a time. So for the example developmental mechanism, if it did it correctly, it's going to produce G0.00 in its ID. I want to walk you through our experimental design. So again, this is our golden data set, that is craft. So we have 3 experiments right here. One is the first step using conditional random fields. That one is evaluated on 10-fold cross-validation. And the second one is our mapping, mapping the textual mentions. So we split our training into 80 and 20% split. So we didn't do cross-validation because our model is really complex. So the input right there is our the gold textual mentions. However, on our third experiment, we connected both of those. So for the third experiment, because we have only 67 full-text articles on the public release of craft, we split those randomly into 60 for training and 7 for testing. So my results are, for all of them, I have results separately. So let's start with the first step. That is the span recognition using CRF. So right here, those are our ontologies. Again, this is on craft version 1. But I have it for all the data set as well, because I wanted to show you how we compare with existing state-of-the-art. I'm working right there with the first version. So we have evaluations here in precision recall and F-score for exact match. When I say exact match, that means did you correctly identify the left and the right side of the terms? However, we have overlap as well. So generally in the results, depending on the ontology, the CRF is not really a difficult task. So, I mean, just using, this is from existing CRF out of the shelf. We didn't really do that much of feature engineering, but we got recent results, especially like for sequence ontology KB, the cellular component and NCBI taxon, we have pretty good results. However, for protein and molecular function, those are a little bit lower. So for the other subtask where we have a golden textual mentions and we did the splitting, within the mapping. So here are the results. So right here, I'm showing you our results for the 3 data sets. Again, this is Craft version 1. So the main difference between version 1 and version 3 is the quality is improved. That's at least what our ontologists say. However, within the version 3, we have for the OBO classes and the extension ones that I explained to you. When we take, as far as the results for version 1, our results are pretty good, again, for sequence ontology, GOCC, and NCBI taxon and cell. However, the others, they're a little bit lower. And generally, results are better for version 3. So in version 3, for example, for GOMF, we went to 0.97. Part of the reason they kind of make it simplified in the annotation. But this is solely given GWAL textual span, can we do the mapping? So from here, you can tell that our sequence mapping is doing pretty good. However, I wanted to show to you both task connected and how we stand with respect to state of the art. So here I'm showing you on the first, on Craft version 1, because we don't have, the others are newly released, so there is no published work on the other versions. Bold here means higher. So these ones are in F1 score. So as you see, our model is doing really, really good compared to others. Except for protein. Proteins are really, the proteins are very short. So normally the mapping doesn't really do good if our textual mentions are short. I have seen textual mentions like 1 character, 2, or 3. So I have done error analysis to show how the system does with respect different length. So that's why we're not doing good for protein, but for the others we're doing great. So again, on this slide, I have the end-to-end results for version 3. Again, I don't have any baseline to compare with, but these are the results that we have. So these ones are in exact match. I should mention, even on the previous slides, all our evaluations are exact match for span detection and exact match for the mapping, if you find the exact ID. If you miss even by 1, that's wrong. So here for Craft version 3, our results are really good, but right there for molecular function, you'll see 1, because we have only few terms, and we're doing good on those. So that's why it's 1. But when you compare both data sets, we're doing really bad when it comes to the with the extension classes. For example, sequence ontology, it goes down, and even KB, it goes really down. So we worked on error analysis. So what we did is I went back. So okay, so we looked to the error analysis with respect to the extension class. So what we saw is there are terms that have very long ID. So for example, we have seen terms like this, terms that has this ID. So if you, for these ones, if you see our system predicts a little, pretty good, but kind of miss it in some of the characters. Remember, we are predicting the individual characters. So here we go, that's the goal, that's what we got. Pato, AXT, and that, but there's some errors right there. So what we did is we replaced these long strings with a little bit shorter and sequential number for all the extension classes. And our results went really higher. So for example, for molecular function, these are the results before doing anything for the long sequential IDs, and these are after modifying. Again, these are results in micro F1. So our results are really good. Again, this is for the second step, just for the mapping, but I have to show you the end-to-end because that's really important. So here are the end-to-end results before and after modifying those long IDs for the extension classes. So as you can see, bold here is higher, and we are doing really good, except for molecular function where it didn't change. It was 75 F1 score, and it's right there. So we did lots of error analysis. The first thing when I showed this to people is what they ask is, okay, does it generalize? So what we did is we did a variance versus bias error analysis to see if there is an overfitting or underfitting. So this plots are actually for GOMF, that is where we do low compared to the other ontologies. So here I'm showing you 4 plots because I'm measuring for the accuracy of each of the characters, and I'm showing you the last 4 because the others are easy and I can get accuracy of 1. So, but the take-home message from this slide, the accuracy for the training and testing is consistent, and there is no overfitting. So I have done this for all ontologies. I'm showing you for molecular function, and again for cellular component where we are doing good. So here the accuracies are higher both for the training and the testing, and for the last 4 digits. So we have done this for all ontologies, and we got pretty consistent results. So the other error analysis that we did is how do we do like for different lengths, for different lengths of input, for example, for short terms or for long ones. So the accuracy here is in green. So what we found here is we are doing good for intermediate lengths. So right there is terms that have only 1 character, 2 character, and even it goes up to 19. So for all ontologies, the results are consistent, and we go really good for not too short, but not too long, but for intermediate. The other error analysis that we did is how do we do for underrepresented or for fewer terms in the test set or in the training set. So right here, I'm showing you the percentage of distinct conceptual counts, and what we found is, again, this is on the test set, okay? So what we found is our results are better for the ones that are occurring most frequently, which is what we wanted. And the results are all consistent for all ontologies. So this is our last error analysis for the end-to-end system. So what we did here is we wanted to know whether we have higher error rates on the first step or on the second step. So red here is error. So we have false negatives here from the first step. These ones are message span. And our first step could say is positive, but it could be false positive or true. So these ones will go to the second step. But the take-home message, what we found is we have higher errors for the message spans and extra spans, which are obviously from the first step, not really from the mapping step. So based on this error analysis, we continue to improve the first step. We replaced the CRF with a deep learning recognizer. I don't have the results here, but that is for kind of next talk, another conference. Our conclusions here: we can do biomedical concept normalization using sequence, and the factors that affect the performance, length of textual mention, lexical variation, obviously. Those are the main conclusions, and with that, I can take questions. Thank you. Okay, Robert.
Okay, this is a very interesting work, and thank you. Great presentation. The question I have is, what percentage of the mentions and concepts in the test set are also found in the training set? And the reason I'm asking is because if the system isn't 100% trained, which I think it is, how does it learn an arbitrary mapping on something it's not seen?
Right, so the system is not trained on 100% of our data set. We split it randomly into 20 and 80. So I wouldn't say all the test sets are available on the training, but to answer your question, I didn't really have the numbers, like what percent of them occur on the training. But that is a good question. That is something that we should take a look at.
Thank you very much.
Thanks.
Interesting
Robert.
work.